My name is Linda Liebert. I'm the Executive Director for the Healthcare Services Institute, which is a division of ISSA. And I'm also the ISSA Healthcare Lead. With me today is Barbara Strain. Barbara is one of the HSI Advisory Council members, and she's also a principal for Barbara Strain Consulting. Welcome, Barbara.
Thank you, Linda. So happy to be here.
Today, we're going to cover the 7 aspects of surface and product selection in healthcare, a comprehensive approach. Our learning objectives are we're going to establish a process and guidelines for the selection of surface materials and products. We're going to learn how to engage a diverse group of healthcare professionals in the evaluation of surface materials and products to support expected life cycle expectations. We're going to analyze potential obstacles to cleaning and disinfection based on the environment of care. We're going to evaluate your facility infection prevention guidelines and talk about where you find those and manufacture IFUs. And finally, we're going to empower you as the audience to ask key questions during the evaluation to reduce repair, maintenance, and make sure the products can actually be cleaned and disinfected without damage. So this is our graphic. In the center is cleaning and disinfection. And I believe we all can agree the most important thing is being able to clean and disinfect not only our environments, but the products we put in our environments effectively. We also can probably all agree that that is a huge challenge, and we'll talk more about that throughout the presentation. Around the center, so the 7 aspects include the clean and disinfection in the middle, but around the outside, location, microbiology, assembly, surface materials and textiles, human behavior, and manufacturer IFUs and warnings are all critical aspects of evaluation to make sure that in the end, you are able to effectively clean and disinfect your product. And we'll go through each one of these strategies. Barbara, can you get us started?
Great. Thanks, Linda. One of the things we wanted to make sure is how do you have effective decision making when someone wants to introduce a new piece of reusable equipment or other product that needs to be cleaned and disinfected? So we, as value analysis professionals, have used a process that was developed since the 1940s around a variety of various industries where we express what the outcomes are versus the cost to achieve those outcomes, which is depicted in the graphic next to you. So value is all that quality divided by those costs. So we put together a multidisciplinary group of subject matter experts, and we bring them together to go through an evidence-based decision-making process. We validate the need and current states and identify gaps. We have a gather data, clinical, financial, purchasing, etc. We analyze that data, contracts, do evaluations. Then we have decision and implementation of those discussions. And then we monitor and assess original goals to reach our total value, which is important. Value analysis, as you might imagine, has a lot of checklists, and one of which is IFUs and other information about reusable medical devices. So when we're looking at that, CDC defines reusable medical devices, and we ask that key question at the beginning of any electronic or internal form or process: is it a reusable device? If the answer is yes, then we want the cleaning and disinfection instructions for use first so we can get started on that review. We involve the variety of stakeholders you see here, from infection prevention, biomed, environmental services, facility managers, so that we're going to all get on the same page about the IFU should we select these devices. And then we're going to look for recalls and other things in databases. So these are just some examples of reusable medical devices. Linda, take
So
it
I
away
want
with our first aspect.
Yeah, thank you. One of the first aspects when you're looking at now, I'm also going to talk about the healthcare environment and the surfaces in that environment as well. So location, location, location is so critical to really understand. There are many different environments of care, and the cleaning and disinfection in those environments are very different quite often. So just for some examples, I want to give you, some good and bad about these areas. First, emergency department, high traffic, high contamination, quick room turnover times. You want to make sure that the rooms are designed to be able to meet those quick room turnover expectations. So the photo in the upper right is actually an outside of the hospital, outside of an emergency room bay. And if you look at that, now high contamination, highly used, but when we look at what's put there, there are a lot of seams, a lot of different textures and textiles and surface materials, all needing to be cleaned, which will reduce the amount of time in turning over an area quickly if it's contaminated. Furthermore, you can see in the ED the human body fluids on the floor and how that gets moved throughout an area, how quickly that would need to be cleaned. And then we've got a couple photos of operating rooms. One, we've seen a lot of photos of the nice pristine room, but in reality, the ability to turn over a room and the need to clean everything in this bottom right photograph can get pretty taxing. And usually in an operating room, they shoot for 9 to 10 minutes between cases for cleaning them. They'll send a team in. They'll do a terminal clean at the end of the day. So again, two different cleaning techniques, but it all matters what the environment requires for room turnovers. So as you're looking at products, for instance, the trauma room here, in that one area, there are over 40 instructions for use that need to be followed in order to turn this area over. There are 20 minutes between that case and the next case coming in trauma that the cleaning needs to be met. So when you're looking at products and the environment, you want to look at, can we actually turn these areas over in the amount of time that's expected using the disinfectants that are required to be used without causing any damage? Could this be a problem? Now, this is a med surg patient room. Again, different environment. There are over 60 different surfaces. The room is cleaned within 10 minutes per day. And who becomes responsible for all of the cleaning? Well, it's not just EVS. And are we using surface materials and products that can be easily cleaned and disinfected in a short period of time? And how often do you have to clean this area? It depends upon the level of care. Real life challenges. So when you're trying to turn an operating room over in 9 to 20 minutes, you can see the damage that is actually happening to, for instance, the pole, the base of the IV. Well, actually, there's instruments on that pole as well. But you can see the damage that occurs when they're trying to clean rooms quickly and the efficiency is not quick enough. So the key here is to understand the location that you're actually purchasing products or materials for, understand the cleaning and disinfection requirements, and ask questions. So what are the infection prevention, cleaning and disinfection guidelines for this area? Every healthcare facility has guidelines. Ask your infection preventionist. How often will this area be cleaned and disinfected? Well, when you're turning emergency rooms over, it could be, 5 to 10 times a day. You want to understand that there are multimodal requirements, so make sure it all works together. How many times can one product be cleaned, and can the product meet the rigorous cleaning and disinfection requires of this location? Barbara.
So we're going to talk about another aspect called microbiology, which is a very unseen world and biofilm formation. How did we get here? Well, the advancement of medical practices, procedures, devices, treatments, multi-drug resistant organisms, hospital-acquired infections. 1 in 31 hospital patients today acquire one of these infections. And it all gets down to cleaning, disinfection, and sterilization agents and methods. So over the time, we've developed a lot of uh, resistant and acquired microorganisms. On the left is the 10 most common pathogens that you find. Some will become very familiar to you, Staph aureus, etc. The other side is 16% are all associated with multi-drug resistant pathogens. So you have difficult situations from the beginning. So when Linda mentioned location, it's also the location of what organisms might be in your environment, what new things are coming, and how do you prevent them. So when you look at disinfectants and cleaners, and they tell you that they have a 99.99% cleaning rate, that means there's still 100 microorganisms left at the end of that cleaning that can then cause issues in the environment that your patients are in. So you want to get to at least 99.9999%. The reason for that is not only do you have microorganisms in your environment, but they attach on very easily to various surfaces to form biofilms. And a group of microbes senses this surface, they adhere, and then they colonize and produce this extracellular polysaccharide. So that's the sort of bubble around them, which protects them from not only the human immune system, but also protects them against cleaning and also antimicrobials. They then can release and start attaching again. So this becomes a very serious issue in healthcare. Biofilm can spread a lot of different ways, either through drain, which might be wet, which might be familiar with you. Also, there are dry. So if you notice when you do cleaning, there's usually wet and dry cycles. That also helps to affect the formation of biofilm in various devices, as well as the design, the IFU, and the damage on these devices, and overall affects the useful life of the asset that you've purchased. Linda. Oh, this
Assemblies.
is Barbara.
Barbara,
I
this is
forgot.
you. This is one of your favorite spots.
I know. I said, how am I going to do that? Oh, assembly. So here's a really cool thing that you have to think about. Medical devices are made of different types of materials in one product that you might buy. They can't all be cleaned and disinfected the same way because of the different materials. And when they are, then that's where damage occurs. So keep that in mind. We've gone from simple rooms on the left to more complicated rooms, as Linda and I are putting forth for that look like this in ICUs, real pictures. So assemblies are raw materials such as plastics, stainless steel, other metals, acrylics, and they go in to make different things. And then the actual assemblies, you actually put them together. So how are they molded? How are they seamed together? What adhesives are they used? So you've got to look at both raw materials and how they're all assembled to make those assemblies. So if you look at the bed, there's different parts of that, and each of them would need to be cleaned a little differently because they have different materials. In the duodenoscope at the bottom, these have actually been driven out of use in systems because all those tiny little arrows are pointing to surfaces that are many different kinds. And a sort of a grassroots effort by individuals who may have gotten uh, sick by infections where these weren't cleaned well enough. then FDA physically told individuals that these needed to be removed from their environment. So the IFUs, cleaning, location, assemblies, etc., very important to not spreading these diseases. Safety issues also can delay care and they can increase different outcomes. So they can be recalled if they're damaged and other different effects. So people see these a lot. Internal alerts can happen every day. I'm having trouble with my switches. Their paint is faded and I can't read them. Cracks have occurred. We don't have enough equipment because we've taken some out. The equipment was just purchased. How can it be failing? So a lot of these things happen every day in our hospital healthcare environments. And between 2020 and 2024, 25 recalls were issued for a variety of device issues, such as those listed here, and including IFUs. And the variety of reasons here is even just for update of instructions for use, or the IFUs may be posted, but they were never put forth to be reviewed by FDAs. So due to cracks, the pump hardware may be damaged. Cleaning products contain, but not limited to, the following may cause damage. So there's a plethora of information that we'll talk later about the IFUs, but it all affects these assemblies at any given time during any given day.
And when damage occurs, I just want to add this here as well. When damage occurs, you're creating tiny microbial reservoirs where microbes can actually get into those areas and colonize up to a point where it becomes very difficult, if not impossible, to actually continue cleaning that device. And we don't, at this time, there's no research to let us know when should this device be removed because it is a patient safety risk. Materials and textiles. This is such an interesting subject. When you look at the materials that are in a healthcare setting, whether it's on a medical device or it's actually in a room, you've got both soft and hard surfaces. Soft surfaces, according to CDC, cannot be effectively disinfected. And so you're going to find a wide variety of what they would consider sort of not really soft, but not really hard either, to try and combat that. And you see furniture, for instance, that may try and address that so you can clean them. So you have textiles, you have laminates, you have stainless steel. And I just want to stop here for a minute at stainless steel because people have talked to me about, well, Linda, hey, we're just going to put the whole hospital stainless steel again and life's going to be good. Well, wrong. This stainless steel, these two that I've used here, you see frequently in the hospitals. The top photo of stainless steel is brushed stainless steel. Now think about that for a minute. When they brush stainless steel, they have created tiny microbial reservoirs. And I will further add that in the IFUs for stainless steel, bleach is not recommended to be used on stainless steel surfaces because it will corrode it and rust it. So we're back to there is no perfect surface. You have to identify the location on where it's going to be used. And then you have to be strategic about how you use it and make sure that it can be clean and disinfected. Another favorite spot for me is the patient bathroom. Going back to the location discussion and the microbial discussion, you've got a toilet that does not have a lid here. I'm sure many of you have heard about toilet plume. So as soon as you flush the contents in that toilet, a plume will go up into the air. We now have ceramic tile on the walls there, which has grout. Ceramic tile and the grout have different cleaning requirements. The handrails are brushed stainless steel, and we now have introduced, because we've moved to the universal patient bathroom, a soft surface, which is the shower curtain. You cannot clean all of this the same way. And going back to the original discussion, they usually have 10 minutes a day to clean a patient bathroom. And you can see where the challenges start coming in. My favorite other picture is this, the med surg. I shared this with you a little bit ago. 60 different surface materials and intersections in this one area. Again, 10 minutes every day. Now you get more time on a terminal clean, but we need to really look at how many of these areas do microbes attach to and begin to colonize in. And you want to look right into the surface. Here's a list of questions. Can these materials and products be cleaned and disinfected using standard hospital-grade disinfectants and following infection prevention guidelines without causing damage? We have found conflicts in those two things. How often can these products be cleaned? A hospital bed mattress, depending upon the patient need, could be cleaned up to 14 times a day. Will that product withstand that? Ask about no-touch technologies. So, does that hospital also use UV disinfection as well as the manual cleaning? You've got multimodal solutions that are being used. You want to make sure that they're compatible because there is no silver bullet. And then there's the human behavior. We have very little, if no control over human behavior. The challenge actually comes here is number one, hand hygiene is critical. So I did a non-scientific behavioral observation when I was an advocate for a patient not too long ago. And what I observed was all of your healthcare workers would come into the room and pump in. They would proceed to reach into their pocket and pull out a pen, just like they did in the previous room, in the previous room, in the previous room. And then they started touching a variety of other surfaces before they ever touched the patient. It was interesting because it was clear healthcare professionals were trained for this specific behavior because every single healthcare professional did exactly the same thing. Then you have the patient and visitors. I broke the visitors into family and others. Family members instinctively grabbed something to clean the area around the patient, which again, unless they're trained, aren't sure, what to use. And then you have your patient. In this situation, the patient that I was an advocate for was non-ambulatory, and she was only given one opportunity a day to wash her hands. So we have a lot of things that come together with human behavior that interferes in a variety of ways with the ability to effectively clean and disinfect, and as well as movement of microbes and transmission. And so who's responsible and who's expected to clean these areas? Well, actually everybody is. And so one of the gaps that we've learned is that we need to build consistent training programs so that we've got everybody trained around all of these aspects, as well as how to effectively clean and disinfect in any given area. And again, a list of questions. Who can you engage and collaborate with? Infection prevention, etc. Evaluate the training programs and make sure everybody's being trained the same things, at least at a foundational level, so they all understand how to work with equipment and how to clean it. And then finally, educate the why. Why are we doing this? It's to save the patient and prevent them from getting an HAI.
So we're going to talk about the manufacturer's instructions for use and their warnings, and how to read and interpret IFUs for care, maintenance, and warning the manufacturer provides for devices and surfaces. It's really critical that these instructions for use not only be easy to understand, but also how are they written. Next slide, Linda. Thank you. So the EPA, the Environmental Protection Agency, has about 850 registered hospital disinfectants for hard surfaces. You just learned from Linda, there are no disinfectants approved for soft surfaces. Amy has different guidelines for the design and testing and labeling of reusable medical devices and resources for manufacturers of medical devices. CDC has cleaning and disinfecting of environmental surfaces and healthcare facilities guidelines, also stating about following the manufacturer's instructions for proper use of disinfecting. Next slide, please. The FDA is the one that guides in the Title 21 801 section called Labeling Cleaning Instructions. And they're given clear and complete cleaning instructions are the goal of an IFU, instructions for use. In addition to how to operate a piece of equipment, everything, it's how to maintain it, how to clean it, how to disinfect it. So the supplies, step-by-step procedures, how often to clean the device, what cleaning accomplishes, failure to clean and what happens as a result of. And highlighted here are appropriate warnings and precautions for cleaning agents, such as if they pool where they might be connected or around corners, that might damage the actual device. And then describe the results of using improper cleaning solutions or methods. So if the IFU is not followed the way it's presented by the manufacturer, then any untoward damage or other conditions may occur that may make that piece of equipment either not function properly or continue to get damaged by having these micro fissures, microbiology, microorganisms come into play. And then it should also include suggestions for disposal of any of the cleaning agents if appropriate. It's got to be very complete. And examples today are sometimes often lacking some of these or point to other agencies like follow the EPA instructions for this particular disinfectant or cleaner. Next slide, please.
I want to just quickly add something here,
Yeah.
Barbara, because we've got some time. The the Joint Commission has recently set a standard requirement, and they're enforcing it now. It's been there for a while. And that is that you follow the instructions for use and the guidelines from the manufacturers for cleaning and disinfection. And while this is great, what we have found are gaps. And when I talk about gaps, I talk about we have seen, for instance, a IFU where it was for a warming component for a patient in the operating room, and the recommended cleaning and disinfection was Pine Sol. In a healthcare facility, you cannot use Pine Sol to clean it, but it could potentially be a violation of the hospital if they don't follow that without going through an entire risk assessment procedure. And we're going to talk more about that. We're finding the Institute has found conflict between FDA, infection prevention guidelines in a healthcare facility, and manufacturer's instructions for use. And so there's a gap there that we're working to address.
Thank you. So pre-market testing of reusable medical devices by manufacturers is sort of a title about these labeling methods. And right in that standard, it says the compatibility of the device with reprocessing methods and the ability of the methods to successfully reprocess device needs to be validated and stated in the instructions for use. The instructions for use still are lacking because there are issues with not being very prescriptive in the description of what needs to happen in the labeling, as the next slide will help point out. So today, in the instructions for use, the selection of the actual cleaning and disinfection agents is not clearly indicated. The compatibility testing is not clearly defined or discussed. And then durability, if you think about it, When you're cleaning, you might clean something up to 10 times or more a day if it goes in and out of a patient room, like a glucose monitor or some other handheld. But the lifetime of that device might be 2 years, but do they take the durability over all of that time that the frequent uses and cleaning are going to happen? So, how often should that cleaning and disinfection occur? Every time, once a day, at the end of the day, etc. So it all has to be clearly identified. The results, if you follow today, as we know, is potential for damaged equipment. Those inadequate cleaning and disinfection that could lead to microorganisms building up and being transferred to patients, the biofilm production that could be then long lasting, and then that whole spread of microorganisms. We know that that happens today, again, using mattresses as an example. There are many papers out that have been documented by gene sequencing of organisms and others that the same patient being put on the same exact mattress in a patient room, that the previous patient was diagnosed and had a hospital acquired infection, acquired the same exact genomic infection by seepage of the organism that was from the actual mattress. So in the future, what HSI has been working on now for almost 10 years is having a fully tested and validation of raw materials as well as assemblies. We want to make sure that they're demonstrated that they can be cleaned and disinfectant by the most popular or the most well used and effective products used in healthcare. They don't have to be tested against every single one on the market. And HSI understands and know these things and have a lot of experts that we can call on. We also want to assure that stated lifetime of equipment is accurate and without damage. The results of this would be patient safety from healthcare acquired infections, accurate lifetime of devices, which then actually then goes towards your financial investment and how often or how much further that device may last in your environment.
This
Next
is a
slide. Yep.
it's a huge issue that HSI is really working with and working with other organizations as well who also are trying to address the instructions for use. Manufacturers can really partner with us in developing simple guidelines around creating instructions for use, the content that might be missing. And so we have been working and we continue to work on collaborative ways of working with manufacturers so they understand. We've learned that manufacturers at the design phase are not necessarily thinking of how to clean and disinfect it. In reality, the cleaning and disinfection discussion should start at the very beginning and then work its way through to completing an IFU that can really be used. And here we're going to talk about the bottom line. Cleaning and disinfection is the bottom line. One of our biggest frustrations is 2 years ago, I was hired by a company to do 37 different architect and design presentations. And the name of the presentation was Infection Prevention Begins at Design, Strategic Use of Surfaces. And at the beginning of every one of the presentations out of 37, and sometimes there were 5, I did a group up to 75 in these 37, I would always ask, how many of you have read the manufacturer's instructions for use and care and maintenance before you have specified a product for healthcare? I'll let you guess. I was lucky if I would get 1 or 2 hands up, and quite frequently I got none. I also did the presentation at a conference. There were 150 people there. I got 10 hands that went up that actually said They are reviewing this information. We need to start building products and hospitals that can be effectively cleaned and disinfected based on the area that the products are in and based on the type of microbes that we're working with and knowing what kind of disinfectants will be used. There is no silver bullet. There are many different types of disinfectants that are used. There's 7 categories, and there's also no-touch products that are available as well. We need to start looking at cleaning and disinfection as the beginning issue and really start addressing that from now on. And when you do that, you really need to look at all 6 of the aspects that we've talked about: location, microbiology, materials and textiles, assemblies, and the human behavior. Review the infection prevention guidelines, which can differ between each facility. And we want to evaluate how things are being trained when it comes to cleaning and disinfection, and then look for validation from our manufacturers that they have really tested their products so that we know for sure that they can be cleaned and disinfected with the standard products that are being used without causing serious damage. We also want to make sure that we know the expected life cycle of a product. So for a long time, you purchased a bed. Well, they didn't give you the life cycle of the mattress. Now they're starting to state that it's 1 year and it needs to be repaired and replaced. So our thinking really needs to change a little bit as we evaluate products that we're buying for the healthcare environments. And so these are some of the questions that you can ask during the process of evaluation to finally make it a final decision on whether or not this product is going to fit in this location and whether or not it's going to be safe for us to use on an ongoing basis. And we want to give you an example of a case study. Barbara, do you want to start this
I'll
conversation?
start off the conversation. We were excited when we learned about a hospital and a manufacturer willing to remain anonymous but provide us all of the information over a several-year journey about what happened in their organizations and what can come of this. And it really results in the true arc of the 7 aspects of surface and product selection in healthcare. Next slide. So the problem was a Midwestern hospital purchased several state-of-the-art medical device monitoring systems, but within 2 years, they saw visible damage found due to chemical exposure during the disinfection process. So the general gist of the results of this study, which you can find a copy of on the HSI website, and I want to really remind everybody, really go there and look through this and read it, because there's a lot of information, including details about all of the data that was gathered, the dollars and cents it cost everyone. But the general results are the root cause of the damage was chemical exposure to components using disinfectants that were not approved and caused damage. So there was a disconnect between the IFU and what the infection prevention and control guidelines were in the Organization for the Healthcare. And they couldn't reconcile these two. So there were challenges and gaps, and primarily the lack of understanding about the guidelines that the hospital were following. Because of some of their patient population, they couldn't use what was considered the recommended disinfectant. There was also information in the medical device company's IFU in small print at the bottom of their little chart for the IFUs about not using a particular disinfectant, which was what the hospital was using. So there was this communication and lack of information there. And then, of course, that leads to communication breakdowns within the organizations and between both organizations. And commonly, when we do FMEA or root cause or A3, communication is a key thing. So the potential to risk to the patients and high costs for both the hospital and the medical device company could have been avoided if we had started with that value analysis process and everyone was gathered to the table. And the next slide, Linda, I think we also have a couple of things about the gap in surface compatibility, communication, and collaboration. I'm a big fan of alliteration, so compatibility, um, collaboration, and communication were key things there. And the gaps were the medical device company had minimal reference material for the disinfectant compatibility and durability testing over time. They did them, but they weren't looked at for the whole length of time. The hospital did not evaluate surface disinfection compatibility and durability as part of their purchasing process, which they could have done had they had a big coordinated effort with collaboration and communication. And both organizations realized that if they had assigned single points of contact to do this coordination, such as value analysis or other professionals trained to go through a very detailed checklist and involve everyone, then that implementation could have ensured a more formal cleaning and disinfection training actually occurred using the products that were both effective for the equipment as well as met the hospital goals for their patient.
And I'm going to add to this as well. Someone in the chat just wrote that they had sent a piece of damaged equipment in for testing. As the staff complained, the disinfectant caused the corrosion. It turns out that the corrosion was rust because they were overwetting the device when cleaned and not allowing it to dry. It was not damage from the disinfectant itself. So this also goes to the fact that, and this was part of the case study, they had not trained they, meaning the manufacturer and the hospital together, had not trained the staff. And when they actually took the time to observe, so the device manufacturer took some time then when they started seeing damage to observe how people were cleaning and disinfecting the product. And that was also one of the key areas, was that nobody had been trained. So this manufacturer went back and created a one-page resource guide, and then they came in and they trained. And a little while ago, we talked about who all cleans. Well, you have nursing staff, you have, well, not usually physicians, but sometimes physicians, nursing assistants, environmental services professionals. You have a real blend of people within the healthcare facility that are actually going to be cleaning this. And so one of the gaps is not training everybody. And so this is really important that you have a training program and that you follow the IFU during the training program to be able to address this. So that was a huge gap, and we've seen that in more areas, but we did address that in this case study. So where do we go from here? We gave you a lot of information in a very short amount of time, so we do have time for questions. But where do we go from here? So the Healthcare Services Summit is being held on April 7th and 8th. That in Chicago, Illinois. There's information on the HSI website. This is a summit in its true form. We asked 3 questions. We have 15 lightning talk presenters who answer the same 3 questions. And of course, we've done this before, and answering the same 3 questions brought up many different responses. We also have task forces that are currently working. So we've got the IFU task force working on developing a template and some standards for manufacturers. Testing and validation, as Barbara mentioned, we've got a team that's been working for many years on testing. We have 1 patent that is pending. And then ultimately, we will have a certification program where we will do the testing and we will validate and review IFUs. And then we also have an education committee to help us develop programs like this that will definitely meet the need. You can also join ISSA, and we do consulting and a lot of other things. And for any additional information, you can email me. I put my email address there. I noticed Martha did put the event registration in the chat, so you can click on that. And we're happy to take any questions. Barbara, do you want to read the one that just came in?
Yeah, I'll read a couple that came in while we were talking, and what I've done is I've kind of grouped it into one question. So either both of us can take a stab at this, but um, someone had mentioned that antimicrobial additives can be added to products as they're made, whether if it's in liquid form of how the products are made, and as well as other products such as, tiles and things like that. And also, ultraviolet light can help knock down different microbes, as well as air scrubbers and various things. So I thought we could sort of noodle this sort of thing about all of those different things that we've seen before COVID, since COVID. What is sort of the end all be all? And I think you said it well, Linda, when you said there isn't one magic bullet. So I'll take the first little stab at this is knocking down how many microorganisms there are in the environment is a great start.
So there's a couple of things when we look at antimicrobial products, and that is number one, there are antimicrobial stewardship programs in healthcare facility. You must understand that they are not the end all be all. Number two, they can and will build microbes can and will build resistance to those antimicrobial products. It's happening with copper. And so one of the things you want to be sure that that you do is test for resistance. You want to make sure that you look at, are they compatible with the disinfectants? And the other thing is a lot of times there's a false sense of security when it comes to antimicrobial products. And that is that it's killing everything and we don't have to clean it as often as we do. The quickest way to get an antimicrobial product to not be effective is by not cleaning it, because as soon as you get bioburden on that surface and enough of it, it becomes ineffective completely. And I've been in hospitals where I've observed professionals not cleaning a surface, and I asked them about it, and their comment was, "Oh, we don't have to clean that because it's self-cleaning." So need to think about resistance and use those products strategically. And number two, you need to make sure that they can also be clean and disinfected using the standard products in a healthcare facility.
Linda says things really well is when she says that you really need to be able to knock down that growth, and not one thing really can be the end all be all. However, collectively, as we're all working together, we can do a lot, but we need to make sure that, and Linda mentioned it, so I was going to say out of my mouth before I went away was cleaning is so important, because the actual scrubbing with like a micro cloth is very important to knocking things down. You don't know if blood and body fluid that you might not be able to see or whatever has hit any of the surfaces. They might also contain microbes, but they may have only been there for a few seconds or a minute before you came into a room. So it's really critical that we do all of the steps and whatever's comfortable for you, but the cleaning and disinfecting at the end still must occur to be able to do everything well and to assure a safe environment. We know from studies that have been published that when a room has been terminally cleaned, even if it has UV light, which has to touch the surfaces and be able to find them, or having antimicrobials embedded in everything, you can still find at least one or more organisms growing that could then perpetuate before the next cycle of all of that cleaning and things are done. So it's not just we're saying that any of those things are not effective. They are at knocking things down, but we need to make sure that that cleaning and disinfection is done accurately. Another question that had come in was expected life testing. So someone said, we do the expected life testing. And I think that when you're doing it, it would be helpful that when you're trying to talk about a new piece of equipment that could be very life-saving or revolutionary, that those where you're presenting that to, tell them what you did, how you did it, how long you did it for, and what you included in that durability and compatibility testing. And then those questions ensued. Don't just assume that, well, we have an IFU, so it means we did all that. It's much better to talk about what your processes were so that all those things could come out.
There's some really good comments in here.
Yes.
So a So one of them, medical equipment is getting more and more complex for a lot of this equipment, IV pumps, etc. I feel like we need to go back to having the SPD experts clean these items. One of the things I want to add here is what we really need to do is partner with our manufacturers. They're the innovators. They have the ability to design products that will meet our needs. We have a gentleman that sits on our testing and validation team that is right now working with medical device or medical surgical equipment where they have developed some surgical instruments that can both be disposed of and reused. It's fascinating work. But what's happened is the manufacturers are starting to think, okay, we want to make it easy for whatever to care for the patient, but they're not thinking about cleaning and disinfecting. And when we've gone back and talked to the manufacturers, they've actually been able to innovate products that address these issues. So, a lot of that responsibility, I believe, also should be laid on the manufacturers. Team up with HSI, talk to us about so you'll understand that you may design your product to be cleaned in a laboratory, but when you get into a real world setting, it's not like that at all. And so I agree with you here. And I also agree about the special tools that are needed. It's ridiculous that we're actually, reducing ourselves to those special tools. And of course, that manufacturer is the only one that has that tool. I've also been in reprocessing areas where they've had little tiny brushes that they've tried to develop or find so that they can clean out cracks and crevices. Again, this really means we need to also engage our manufacturers with all of this as well. Um, let's see,
Right.
there's
I want to address the question that someone asked about what's a facility to do when the answer is no to many of the questions about how do I clean it, et cetera, because we really need to have the device. So you really have to do a risk assessment. You have to also work with the manufacturer. Could they do additional testing? Do you work together with them to talk about here's our situation as far as we need to use this, we need to use it between patients or on the same patient many times a day as a reusable, but what can we do? So it's that partnership that you really have to form because additional testing may have to happen. You need your risk manager and your quality chiefs to also weigh in on these with your infection prevention and control. You want to do the best thing for the patient, but you also want to do the best thing for your entire organization as well. So working together as partners is really the way to go.
I have one here that I want to talk about because he says much of this discussion is on the equipment itself. What about actual architectural surface materials that are used throughout the space, especially in high traffic, high touch areas? Do you have thoughts on the best materials to use? Great question. So I had the honor of working on the Project ER1, which was an all risk readiness emergency department. And this was at Washington Hospital Center. And one of the issues is when you have a trauma or you have an accident or a biohazard, all of these patients arrive at the same time. And how do you actually clean in between these patients? Because they're highly contaminated and you need to address those issues. So what we did with the materials in some of that space is we tested a variety. We minimized the number of seams. They actually used solid surface material on the walls from the ceiling down to the floor, and they coved it into the ceiling and into the floor, number one. Number two, what they did is they used a different color material in the what they considered were high touch areas. So that if someone came into a room, they had a quick turnover time. I wish I had time for pictures here because I've got a lot of pictures. So when a patient comes in, they're done with that patient, they're removing the patient. Somebody that's going to clean that room, whether it's EVS or nursing staff, whoever it is, can recognize the high touch areas by the color of the solid surface that they put into this area. Now, solid surface is non-porous. It's very durable, which is one of the reasons why they chose those products. And they actually incorporated the sink into the wall. So it was there were no seams. And so they recognized high touch by a change of color. They recognized durability and ease of cleaning by using a solid surface material. It was fabulous. And they did testing. The area that they had trouble with, which was really interesting as we were going through doing the evaluations and removal of microbes and how quickly can you clean. And there's all sorts of write-ups on this. But it was very effective in providing the least amount of assemblies of materials in a room, the ability to quickly turn a room over, the ability for someone to see where the high touch surfaces were and be able to keep patients safe. And so that's just one example. I have many, many, many more examples of things that architects and designers have done. But again, it goes back to using firms that have experience in healthcare, that understand the needs of the location and what they need to do in these areas to be able to make it easy to clean and disinfect. So I thought I'd answer that one.
Yeah, I'm going to take the last one. The Joint Commission says we cannot risk assess away the law, which are the IFUs. And we're not suggesting you do that. However, working with the manufacturer means they do more testing and they update their IFU. So that does become what you follow, not just I have an issue, so we're just going to, try to risk it away. You need to involve all your villages together so that around our 7 aspects, so we can make the most reasonable, safe environment for our patients and follow all the regulations and guidelines. So those IFUs would have to be updated as well.
And one more quick thing. I mean, this is a great discussion. The IFUs, one of the things that we have learned, and we learned it in the case study, is, in many other situations, is that there is a big conflict between the number of IFUs in any given room and the expectation that you follow every single one of those IFUs and make sure that things are being cleaned and disinfected in the way that they're supposed to be done. And you saw the trauma room where there are over 40 different IFUs in that room. That is an absolute impossible task. And we need to train or educate our regulatory agencies about what actually happens in a healthcare scenario. You've got 20 minutes to turn a trauma room over. You've got 40 different IFUs. Not all of them can be cleaned and disinfected the same way. We need to go to the table and talk about how do we really address this? Because this is not just a hospital problem. It's a manufacturer problem, it's a regulatory problem, and it's a patient safety risk. And I will step off of my soapbox because we are right at noon. I want to thank everybody so much for joining us today, and stay tuned for more in the future.